Today, Year 11s, we're going to look at exchange, cellular exchange. Specifically, we're going to be looking at diffusion and osmosis. But before we do that, we need to explain a couple of features that affect both diffusion and osmosis. Obviously, diffusion and osmosis, as we've just learned from the practical you just completed, that's driven by a whole range of different factors. Diffusion is the movement of substances across a membrane, typically, from a high concentration to a low concentration. There are a range of factors that affect this, and we'll go into each in quite a bit of detail. But the first, because we're considering cells, is cell size. A small cell is more efficient than a larger cell. And there's quite a few different reasons for that. The basic reason is that the rate of exchange, the speed at which a molecule is able to go from the outside of the cell, so from the extracellular fluid, into where it needs to go, is faster in the smaller cell than in the larger cell. And we can use a very simple example of this. If we've got a molecule on the outside of this cell, and its target, which I'm just drawing green, is right in the middle, the distance that it needs to travel is less compared to if I have the same molecule on the outside of the larger cell and it needs to travel to the center of this larger cell. The distance traveled is greater. This just means that when we're going to exchange substances, a smaller cell has an advantage over a larger cell. Now, this has meant that evolutionarily, from an evolutionary perspective, smaller cells are selected for. Smaller cells are more likely to survive. As the cell size increases, the rate of exchange increases. And this is related to the surface area to volume ratio, or we shorthand that to SAV, where what we're after is a high surface area to volume ratio. We want more surface area per unit of volume. So this needs to be high, while this is kept as low as possible. And smaller cells are able to do this. In class, in our next lesson, we're going to use a couple of examples using a ping pong ball, a tennis ball, and a basketball to show you exactly how that works. But at the moment, you're going to need to take that on fact. If you understand the mathematics behind it, perhaps you'll get a better grasp of that. And that's what we're going to focus on in that example. So the key takeaway from this is that exchange at the cell, at the cellular level, is faster, is more efficient for a smaller cell. We'll talk about that more when we use our demonstration. The other thing as well is to consider shape, the shape of a cell. Most cells, we sort of think of cells as spherical, as spheres, but they don't necessarily always have to be spheres. Now, shape also affects surface area to volume ratio, and we can talk about that. If there's an example or an image of that in your slides, which you can look at, but I'd much rather use an example. So the easiest example is when we're considering something like an ice block. So an ice block, when it's frozen, occupies a certain area, a certain space, and a certain volume. So the volume of the substance, the volume of the ice block doesn't change. Let's say that somebody at some point, they're eating that ice block and they're quite enjoying that ice block, but they drop it. And they don't pick it up because they're embarrassed that they dropped it, they run away, and the ice block melts. And so it spreads out. So we start with this idea of this confined ice block, and then as it melts, the size increases because of this puddle. So we have our volume, and let's say it's 100 centimeters cubed, so this is a terrifically large ice block. But then we have a much larger volume. It's occupying more space. It's occupying the same space, but it's got a much greater surface area. So here the surface area might be 10 centimeters squared. But here it's obviously much larger. So maybe it's occupying 100 centimeters squared. This means here our surface area to volume ratio is 10 to 100, or 1 to 10, which is a high volume compared to its surface area, but over here, our surface area to volume ratio is 100 to 100, or 1 to 1. So we have a much higher surface area to volume ratio. The volume remains the same, but just by changing the shape, we can have a drastic impact on the rate of exchange. And to continue our example of diffusion, if we use our ice block, it's more likely that somebody is going to step in the large puddle of melted ice block than it is if they're going to step in a frozen bit of ice block. So shape, as well as size, has an impact on the rate of diffusion. The closer or the more spread out a cell is able to be, the more flattened and the more space, the more surface area it can occupy, then the faster diffusion will be. Because individual cells must exchange substances with their extracellular fluid, this surface area to volume ratio is incredibly important. They are after as high a surface area to volume as they can possibly get. Because we are multicellular organisms, we have several problems with this. We are big. We don't occupy terribly much surface area, but our volume is quite large. We are large organisms. Whereas unicellular organisms, they've usually got a significant advantage here, which is part of the reason why bacteria still exist. They're able to exchange materials with their environment incredibly rapidly, whereas we have to have specific features for that. And part of the test you'll need to go through leading up to the test, part of the skills that you'll need to develop, is working out how our organ systems or how our cells have adapted to allow this to occur. Well, we're looking at movements across membranes, so I'll need to get rid of this in a moment. In order to control our extracellular and intracellular environments, whether you're unicellular or multicellular, cells must exchange substances. In order to get things into the cell, so in order to import things like gases like oxygen, glucose, so nutrients, and minerals like iron and calcium, we actually need to get those from our environment. Our cells can't make those, particularly animal cells can't make any of those. So the movement of substances across membranes is dependent on several conditions. 3 of them we'll consider here. The surface area available for exchange, so that sort of relates to our surface area to volume ratio. The nature of the substance being exchanged, so that the substance itself, and then the composition of the membrane. Remember in our previous videos that the membrane is made up of a mosaic. It's got our phospholipids, it has carbohydrates, it has proteins, it has cholesterol. So all of this composition of the membrane determines what can come in and what can come out. Now that's what comes in and what comes out, what is able to be exchanged. There are also factors that affect the rate of exchange. So things like concentration gradients. So how much of a substance is outside of the cell compared to how much is inside of the cell. Because with concentration gradients, the steeper the gradient, the higher the concentration difference, or the greater the concentration difference, the faster it's going to occur. And then temperature is also going to affect it, because what we're dealing with here, diffusion is the movement of molecules. Higher temperature means that molecules are moving faster. So higher temperatures mean that we have a greater rate of diffusion. If we have just some examples, so this is just to show concentration gradient. We've got our graphs here, and let's have 3. So we've got our 3 graphs, and along here we have concentration on the y-axis, and on the x-axis, let's say we'll have the location of where it is. So we'll say ECF for extracellular fluid, and then cytoplasm. So we have 3 different cells found in different places in the body. Doesn't really matter where for the moment, but they're found in 3 different places. Now, in the extracellular fluid in this case, it's relatively high concentration. Cytoplasm, low. So we have quite a steep gradient. So the rate of diffusion is going to be quite high. Whereas here we have a slightly lower concentration than ECF and slightly lower concentration in the cytoplasm. And then here we've got a flat line. So we have fast diffusion occurring here because of how steep that gradient is. We have a moderate diffusion occurring here. And then we have no diffusion because there's no gradient. The concentrations are the same in the extracellular fluid and the cytoplasm, so we have no diffusion. Our rate of diffusion is 0 when we have two sides that are equal, when we have an equilibrium, we call it. When we're looking at the nature of the substance, so this is when we're talking about rates here with concentration gradients, when we're talking about nature of substance, we need to know some really key things. The phospholipid bilayer, the cell membrane, which is our phospholipid bilayer, that is made of predominantly fats. So this means that because it's fat, because the cell membrane is mostly fatty, it's going to repel water and water-soluble compounds. So although water is a very small molecule, it's able to penetrate, but lots of water-soluble molecules, so polar molecules, very large polar molecules, and some ions, or all ions really, are not going to be able to get into the cell membrane. So this means that those substances which are still required by the cell are going to have to go through other paths. They're going to have to use those proteins that are dotted throughout the cell, those protein channels. So lipid-soluble substances, things like alcohol, fat-based substances, so steroids that the body makes or steroids that we inject into our cells, they're going to be able to get directly into the phospholipid bilayer. They're going to be able to enter the cell because they're able to dissolve. They're able to dissolve through the cell. Extremely small molecules, so water, urea, gases, they're able to pass through very easily because they can slip between the phospholipid molecules. Anything that's small and uncharged is able to do that. Larger water-soluble molecules, so things like amino acids and sugars, they have to pass through these special protein channels. So the nature of the substance really determines what can come in and what can come out. Remember, it's those 3 dot points there. The nature of the substance is one of those that determines what's coming in and what's coming out. When we reach out, something that I mentioned very quickly before, here, on this far right or far left, depending on how you're viewing it, this furthest graph here, we have reached dynamic equilibrium there. So I'd best write that up. We're at balance. This means that there is no net diffusion. Molecules are still moving because we're above 0 degrees Kelvin. The molecules are still moving around, they still have energy. So it's dynamic because they are moving, but there's no net movement of the molecules. So we're not getting this big change in the concentration gradient anymore. And in class we'll spend a bit of time looking at some examples of that. The movement of molecules across a membrane with the concentration gradient is what's called diffusion. We're going to look a little bit about osmosis and we're going to see that in a moment. But diffusion is a passive process, which means it needs no energy. The energy comes from the concentration gradient itself. These molecules are trying to achieve equilibrium. Diffusion can only occur as long as the membrane is permeable to the solute, as long as the membrane is actually able to accept or allow the molecule to pass through. So there's a couple of questions to cover there and we'll go through some activities, but there's those 3 things. Movement of molecules across a membrane is called diffusion. With its concentration gradient, it's called diffusion. Diffusion requires no energy, so it's a passive process. And diffusion can only occur if the molecule is permeable to the membrane, if the molecule can actually cross the membrane. Now we're looking at facilitated diffusion now. So we'll draw a rather large example of a cell, just to make life easier. And we've got So here we've got a protein channel. Bit in blue is our phospholipid bilayer. So this is our cell membrane. And in green we have some solids. Let's say that these are sodium ions. Now, sodium is charged. It can't, and it's water soluble, so it cannot get through the membrane. So sodium is impermeable to the cell membrane. But the cell needs it, so it has a protein channel. Therefore, it must use a channel. And here is our channel. So the sodium is then able to cross into the cell. You'll notice as well there is low concentration of sodium inside the cell and high sodium in the extracellular fluid. So our sodium molecules are going to go into the cell until they reach dynamic equilibrium. But they need to use protein channels. When a substance uses a protein channel, we call it facilitated diffusion. And the reason we call it that is it's because the protein is facilitating the diffusion. But it's still diffusion. It's still a passive process. It requires no energy and can only go down the concentration gradient, which means that this will stop as soon as we reach dynamic equilibrium. This can be a problem for many cells. We'll talk about the solution to that active transport later on. Now we're talking about passive transport. It's movement of molecules with their concentration gradient, down their concentration gradient. There are a couple of really important features of these protein channels though. They are very, very rapid. A protein channel that transports water can transport 1 million molecules of water a second. But they're also specific. So remember we said that the nature of the membrane, the composition of the membrane, determines what can go across. Protein channels don't just allow everything across. They're specific. So this protein channel is a sodium protein channel. It is specific for sodium. And glucose protein channels are specific for glucose. And calcium protein channels, etc., etc. They are specific for what they are able to allow across. This maintains this semi-permeable or selectively permeable nature of the cell membrane. But channels can also become saturated. So channels can get to the point where the concentration outside is so high that they can't pump any faster. They can't allow these to go any faster. If you imagine it's like a bridge. So Sydney Harbour Bridge on any sort of morning is going to be having a traffic jam. And that means that although the bridge is open, people can't move any faster because there are just so many cars on it. It's the same with the protein channels. We have this open network, but eventually it can't fit any more in. It's going to be pumping as much sodium as it can into the cell, but there's going to come a point at which it can't work any more efficiently. The other thing as well is because it's a protein, it can be inhibited. So certain drugs or certain compounds, toxins or poisons can actually block the channel. They can come in and take the place of the sodium or glucose or calcium or whatever, and they can stop transport happening. When we have diseases, when we have sicknesses like diarrhea or even cystic fibrosis for people who have that condition, that's actually a condition where the protein channels that are supposed to remove and allow things to go in and out of the cells aren't working. So in the case of diarrhea, it's where we're supposed to be having salt pumped out of our intestines so that water will follow it, and those protein channels are blocked. And so they're not able to pump that sodium across, so the water stays in our feces and it comes out the other end as a rather unpleasant sort of experience. But that's what's happening there. So proteins, because of their nature, because of their specificity, they can be blocked, they can be inhibited. But remember, it's a passive process, so no energy is required. Now we've got to talk about osmosis. Now, osmosis is diffusion of water, but because water is what everything is dissolved in, the cytoplasm is mostly water. The extracellular fluid is mostly water. If you're a fish, your entire external environment is mostly water. So we need to treat water a bit separately. So we discuss water and have some different, not different rules exactly, but we have some different terminology for water. So I'll just put up our heading of osmosis here. Now, osmosis is still a passive process. It still requires no energy. And it's the movement of water molecules across a membrane. And the membrane has to be selectively permeable, and we'll talk about that soon. Now, when a solute's in a solution of water at least, so here is our water, this is our container of water, and we add something into it. So let's use, because it's easy, so we're going to be adding sugar molecules. So we add sugar into there. Now initially all we had was water, but now we've added sugar, and the sugar has dissolved in the water, which means that these sugar particles, which I'm just representing in blue, are surrounded by water molecules. So where all we had before was just water and all of these free water molecules floating around as happy as they could be, now they've got to occupy sugar molecules. Now, there are still going to be plenty of free water molecules floating around the place that aren't bound up with any sugar. So there are still some single water molecules that are free and that are available there. But because we've dissolved our sugar into there, we have less of those free sugar molecules. That's our starting point. A pure sample of water will not have anything dissolved in it. And so the water molecules are completely free to wander around. As soon as you add something in there, those water molecules are attracted to that substance if it's soluble in water. They're attracted to it, they surround it, and they dissolve it. And this reduces the amount of free water molecules, so it stops the water from being able to move, or it reduces its ability to move, because there are less free water molecules. That's our starting point. If we throw into that same situation, so we'll draw our beaker again, but we'll make it slightly larger. If we put in our selectively permeable membrane, so our selectively permeable membrane, and it is impermeable to sugar. So sugar cannot pass this membrane. And we've got our water sample here with our free water molecules on the left-hand side. So this is our pure water. But over here, we have our water sample with its dissolved sugar. Those blue dots again representing that sugar. Again, because it's a diagram in science, we have to label it. So these are our water molecules. Here are our sugar molecules. Need to draw a few more because we will have some free water molecules here. Now, we've got these sugar molecules. They are taking up those free water molecules now. So we have an imbalance. There is a concentration gradient here. There is lots of sugar, so we'll draw that in blue. Labels first. We need to label our axes. So left-hand side and right-hand side. So on our left-hand side, we have no sugar. And on the right-hand side, lots. So the sugar would like to go into the left-hand side according to its concentration gradient, but it cannot. It cannot cross that membrane. If we look at our concentration of water, lots of free water molecules here on the left-hand side. Not quite so many on the right-hand side. Now the barrier, the membrane, is not impermeable to water. So water can travel down its concentration gradient, and it will. These water molecules will move into this right-hand side of this beaker, and they will diffuse across, and that is the process of osmosis. So water molecules move down their own concentration gradient, and we call that the osmotic gradient. The osmotic gradient because it's what causes osmosis. And the force with which it does that is called osmotic pressure. That brings us to the end of our slides for today. So there's a few activities we need to do in class over the next few days before we have our test. But that brings us to the end of the lesson today.